Hi folks, and welcome to National 4-5 Photosynthesis topic. So for National 4s you'll be studying key area 6, which is cell biology, photosynthesis. Okay folks, so for National 4, these are the key areas that you need to cover. So you need to know that plants need light, water and carbon dioxide gas in order to carry out photosynthesis. And if any of these factors are low or missing, the photosynthesis rate will be slower or limited. And if we overcome these limitations, we get faster growth rates, because if plants photosynthesize, that allows them to grow. So plants carry out photosynthesis to make their food. Now we should be aware of this from Unit 1 because plants have chloroplasts and that is how they make their food. So oxygen is produced and released as a byproduct of photosynthesis. Light energy from the sun is absorbed by a green pigment called chlorophyll and used to make sugars, which store chemical energy. Now what we have here is a photosynthesis equation. So we get carbon dioxide. When that joins with water, so when these two compounds join together, we make sugar. So if plants have carbon dioxide and water joined together, then we make sugar and we get oxygen gas as a waste product. Now what you'll see is light energy absorbed by chlorophyll is required in order to join carbon dioxide and water together. And I liken this to baking a cake. If I want a chocolate cake, I've got to put ingredients together, but I need to mix them, and that requires energy. So it's not just a case of having carbon dioxide and water together, you have to have energy to really push them together. And that energy comes from light, from the sunshine, and is absorbed by chlorophyll. So a wee summary here. If I've got light energy trapped by chlorophyll and chloroplasts, then I'm able to join carbon dioxide and water together to make glucose sugar. And we can see oxygen is given out as a waste product. So the rate of photosynthesis is affected by the following factors: the concentration of carbon dioxide, the availability of water, and the light intensity. It's also affected by temperature, which is not here. I want you to bear that in mind. Now, obviously, I need carbon dioxide to make my glucose. So if there's less carbon dioxide, we're going to have less glucose sugar. Similarly, I need water. And obviously, the light energy is used to join the compounds together. So a limiting factor is a factor that, when in short supply, can slow down the rate of photosynthesis. Now, what we have here is a graph that we have to analyse about limiting factors. Now, what we have on the x-axis is light intensity and the y-axis is rate of photosynthesis. And we have x labelled, y and z on the graph. Now, basically, the rule is if you're going up at an angle, if you're on the way up at an angle, it's whatever is on the x-axis that is limiting photosynthesis. So if you can have a wee look at the graph, you see the graph going up x, x, x. Any point along that line, you go down. If you're on the way up, you go down, and it's whatever on the x-axis that is limiting photosynthesis. So any of these points, x, 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 it will be light intensity limiting photosynthesis. Now that's because if I increase the rate of light intensity, I can see the rate of photosynthesis increasing, and when I decrease the light intensity, I can see that the rate of photosynthesis is decreasing. So I can see that light intensity is directly affecting the rate of photosynthesis. Easy way to remember this: if I'm on the way up, if I'm going up, I look down, and it's whatever is on the x-axis that's causing the limitation, that is limiting the rate. Now, if I can draw your attention to the line marked y, we're on the level here. There's a constant. Now, if you're at a constant, it can't be light intensity, it can't be what's on the x-axis that's limiting photosynthesis. Because if I increase or decrease that, it stays the same. So see, when you look at y, it can't be light. Because if I go up and down in terms of the light intensity, the rate of photosynthesis is staying the same. So some other factor is affecting the rate of photosynthesis. So the rule here is, if you're on the line, if you're on the level, you look up to the level above and spot what's different. So let's look at y. Imagine we're sitting at y. Let's look at the level above and what's changed in that level above to cause the rate to jump up. So at y, I've got a lower CO2 concentration and a lower temperature. But something's limiting photosynthesis. Let's look at the level above to Z. What's different? At Z, I've got a lower temperature, but I've got a higher rate of a higher concentration of CO2. So could it be CO2 concentration that's holding us back at Y? So the rule is, if you're on the level, you look to the level above and spot the difference, and the difference is increasing the rate of photosynthesis. So my answer to that would be CO2 concentration is limiting the rate of photosynthesis at Y. Now let's look at Z. Again, I'm on the level. So it can't be light intensity that's limiting the rate of photosynthesis at point Z, because if I increase the light intensity, there's no change. If I decrease it, there's no change. So it has to be something else. It's not what's on the x-axis. So when you're on the level, you look at the level above and spot the difference, and the difference is what's limiting photosynthesis. So let's look at Z. We've got higher CO2 concentration but lower temperature. Let's look at the level above. Higher CO2 concentration, so that's not changed, but we have a higher temperature. So at Z, temperature is limiting the rate of photosynthesis. So let's consider the two rules. When you're on the way up in the graph, going up in the gradient, you look down to the x-axis to find the limiting factor. If you're on the level, you look up to the level above and spot the difference. And what is increased in the level above is what's holding back the rate of photosynthesis. So let's try this. So we've got light intensity in the x-axis, rate of photosynthesis on the y. So A. Now remember, if you're A, you're on the way up. Think of yourself going up a hill. So when you're on the way up, you look down. So when you're looking down, you're looking to the x-axis. What's on the x-axis that's limiting photosynthesis? Light intensity at A. Let's look at B. Now we're on the level here. So we know it's not light intensity, because if I increase or decrease light, there's no change. So we'll look at the level above. And what's different? Well, at B, we've got 1% CO2 concentration. At C, it's 2%. So what's holding us back at B, what's limiting photosynthesis, has to be CO2 concentration. Now, if we look at C, again, we don't have a level above, but it's not light intensity. So I'd be thinking, what else do I know that holds photosynthesis back? So it could be more CO2 concentration if we went higher than 2%, or it could be temperature. So a greenhouse can be used to overcome the limiting factors of photosynthesis. This allows plants to grow faster as they're making more food. Greenhouses have artificial light so that photosynthesis can continue beyond daylight hours or at a higher than normal light intensity. The use of paraffin lamps into the greenhouse increases the rate of photosynthesis because burning paraffin produces carbon dioxide as well. So again, what we're trying to do here, if I want the plants to produce lots of glucose and get a higher yield, help them grow faster and bigger, I need to provide them with raw materials for photosynthesis. So I need to provide them, for example, with carbon dioxide. I need to provide water. I need to provide an appropriate temperature, and I also need to think about my light intensity as well. Now, the Elodea bubbler is a method used to measure the rate of photosynthesis. Elodea is an underwater plant. And I said in an earlier slide that plants during photosynthesis will release a byproduct or a waste product, oxygen. Now, by doing that underwater, we can actually count the oxygen bubbles and measure the bubbles over time, which allows us to measure the rate of photosynthesis. Now, this experiment's been set up to measure the effect of increasing or changing the light intensity on the rate of photosynthesis. So if you look at the lamp, we can move the lamp further towards the Elodea, and that will obviously give us more photosynthesis because we're increasing the light intensity, so I should see more bubbles over time. If I move it away from the Elodea, I should see less bubbles because I'm decreasing the rate of photosynthesis. Now, I want to draw your attention to the transparent heat shield. Now, if I'm investigating the effect of light intensity on the rate of photosynthesis, that is my independent variable, and that's the only variable that I'm changing. All other factors must be kept the same for my experiment, my investigation to be valid, to be fair. Now, the transparent heat shield allows the light to get to the Elodea, but not the heat from the light. Because if you imagine moving the light closer to the Elodea, without that heat shield, that would be you increasing the light intensity and the temperature. So you're changing two variables. And again, your investigation will not be valid. So we use the transparent heat shield to make sure that the only factor we're changing is the light intensity. Now, we use dilute sodium bicarbonate in the solution in order to provide the carbon dioxide for the Elodea, because remember, we need carbon dioxide and water in order for photosynthesis to take place. Okay folks, we're going to have a look at some of the photosynthesis skills that you're going to need to be able to do. So accuracy. Now, accuracy relates to the measurement of mass or volume, for example. So it's about a measurement. A smaller scale would provide a more accurate measurement of volume. Using a small scale syringe instead of a measuring cylinder with a large scale will give a more accurate measurement of volume. So it's all about the apparatus you use to get your measurement. Using thinner capillary tubing will also allow greater accuracy. Now, if you think about that in your kitchen, if you're wanting to measure out 100 ml of milk, for example, if you're making pancakes, if you have a jug, a measuring jug that's got a scale that's larger, it's going to be quite hard to get a more accurate measurement. But if you're using a jug with a smaller scale, you can be more confident that you have actually measured 100 ml of milk. So it's all about the scale that you can use. So it's about the measurement of mass or volume. So, in terms of accuracy, which scale, the scale at the top or the scale at the bottom, would give a more accurate measurement of volume? Which has a smaller scale? The top one. So, instead of simply counting the bubbles when collecting gas, what we could do to improve the investigation is use a scale and measure the volume of oxygen collected over time. So in this past paper question, the apparatus below was set up to investigate photosynthesis in an aquatic plant like Elodea. So we can see, as described before, the light source. We're going to obviously change the light intensity by moving the bulb closer to the Elodea, and we're going to obviously measure the oxygen bubbles over time. Now, how could this investigation be improved to make the results more accurate? So, what we could do is, instead of just counting, why don't we get a measuring cylinder, collect the oxygen gas, and we can see the oxygen collecting at the top of the measuring cylinder, and obviously set a time for that too. So what we're doing is we're measuring the volume of oxygen produced over time instead of counting bubbles. And the smaller the scale in the measuring cylinder, the more accurate measurement of the gas we would get. So accuracy is really about our measurements. Let's have a wee go at exam style practice for NAT 4 and 5. So what is the rate of photosynthesis affected by? What affects the rate of photosynthesis? So we should have carbon dioxide concentration, light intensity, and temperature. They're all required for photosynthesis, so if they're in short supply, our rate of photosynthesis is going to be reduced, but if they're increased, the rate of photosynthesis is going to be increased. Lettuces may be grown in tunnels covered in green polythene as shown in the diagram below. Suggest a reason why the use of clear polythene may result in the lettuces being ready for cropping earlier. So again, we know that you need carbon dioxide, you need a nice warm temperature, and you need water and light for photosynthesis to happen. So what we're saying is, how is this helping? How is this helping grow the lettuces, carry out photosynthesis? So we could say it's adding warmth, it's providing warmth, shelter, and wind protection as well. So we know that temperature, if we increase the temperature, our rate of photosynthesis increases, and therefore we will get greater growth of lettuce. And by using these tunnels, we're increasing the temperature. Air rich in carbon dioxide can be passed through these tunnels. Explain how this would make the lettuces grow faster. But we know that carbon dioxide is required for photosynthesis. So the more carbon dioxide, the more photosynthesis, the more glucose we make, so the bigger the lettuce is. What is limiting photosynthesis at A, B and C? And I'm going to remind you of the rule. When you're on the way up in the graph, you look down and it's whatever on the x-axis because that's what's down below. If you're on the level, you look up and spot the difference. So A, what's limiting photosynthesis at A? Light intensity. Let's look at B. We're on the level, let's look up and spot the difference. Okay, so at point A, we're going up, so we look down, so A must be light intensity. At point B, we're level, so light intensity can't affect B, because when we increase light intensity, there's no change, and when we decrease it, there's no change. We're still on that level, we're still at that rate of photosynthesis. So we'll look at the level above and spot the difference. So what's different? CO2 concentration, because at B it was at 1%, but at C we've increased it to 2%. And remember, if we don't have a level above, we think, well, what else do I know that increases the rate of photosynthesis? So you could say even higher CO2 concentration, or we could say temperature. So that was your cue again. When you're on the way up, you look down, whatever's on the x-axis. When you're on the level, you look up and spot the difference. Okay, here we're going to predict the effect of an increase in temperature on the rate of photosynthesis. So we know that CO2, we know that water, we know that light, and we know that temperature increase photosynthesis. So an increase in temperature would increase photosynthesis. What is limiting photosynthesis at X and at Y? So if you're on the way up, you look down. If you're on the level, you look up and spot the difference. If there's no level above, you need to consider what other factor, other than what's on the x-axis, is limiting photosynthesis. So let's go up X, so we look down and we come down to light intensity. So light intensity is X, and Y, we know it's not light intensity, so it could be CO2 concentration or the temperature. Now we're coming on to Photosynthesis National 5. So, photosynthesis. Green plants are capable of making their own food using energy from sunlight. And as I said, to make anything, you need energy. This process is called photosynthesis. Now think about the word photosynthesis. We're using light energy to make. We're using light energy to make glucose sugar. And when plants carry out photosynthesis, they grow bigger, so their yield gets higher. So, light energy is trapped by a green pigment called chlorophyll, and we find chlorophyll in chloroplasts. And if you remember from the first unit, we know that green plants have chloroplasts, and that allows them to make their own food. Now, in order for you to make your own food from carbon dioxide and water, you need to have energy, and that energy comes from the sunlight. And it's because plants can trap that energy, they can use that energy to make their own food. So they're called producers because they produce their own food. So as I said, chlorophyll is found inside chloroplasts, which are disc-shaped organelles found in leaf cells. So light is used to combine water and carbon dioxide to make carbohydrate, which is glucose. carbohydrate. Now, carbohydrate is carbon, hydrogen and oxygen combined, and oxygen is released as a waste product. So now we're going to have a wee look at the section through the leaf. We're going to look at the leaf and all the different parts. So the first part, we have the thick waxy cuticle, and that really prevents water loss. Underneath that, we have a layer of cells, quite regular shape, which is called the upper epidermis. Now that's a special layer because these cells are transparent, they allow light to pass through, and we need light to pass through to reach the chlorophyll. And then we have a very special layer of cells underneath that, and they're again very regular in shape, quite like wee soldier fingers, and they're called the palisade mesophyll. And I like to say, what is a princess drinking a palace? What lemonade, but palisade is a way of remembering that. Palisade mesophyll. And these cells are full of chloroplasts. And we can imagine the light coming through the upper epidermis, which is transparent, hitting the palisade mesophyll cells. Below that, we have sponges, which is spongy mesophyll. And again, these cells are full of chloroplasts, so they can also carry out photosynthesis. Now, amongst the spongy mesophyll, we have air spaces because they're irregular in shape, the spongy mesophyll, and that creates air spaces. And that's important because I want gases to come in and out the leaf, and they have to have the space to move, to diffuse. And then we have the lower epidermis. And the lower epidermis contain guard cells, which also have chloroplasts, and stomata or stoma. Now, stomata is singular, stoma is plural, and that's just a pore, just a hole, and that allows gases to enter and to leave. And the guard cell can control the size of the stomata, so it can make it bigger or smaller, or actually just close it over. And then we have the vein, and that's very important because it contains cells called xylem, and the xylem bring up the water which is required for photolysis, for the splitting of water by light. So we need the water up at the chloroplasts, and xylem are big tubes that bring the water up. Also within the vein we have another type of cell called phloem, and any glucose produced in the leaves is transported around the plant by phloem, which is another type of tube. Now, we're going to learn about xylem and phloem in the next unit. Let's have a wee practice and see if we've remembered. So number 1, obviously you've got your cuticle above that. What would that layer of cells at the top be? The upper epidermis. Do you remember what 2 might be? What does a princess drink in a palace? Palisade mesophyll. What about 3? You would take them into your shower with you. Not palisade mesophyll, but the spongy mesophyll. And palisade mesophyll and spongy mesophyll are full of chloroplasts. What is special about the upper epidermis again? Remember, light has to be able to pass through. These cells are transparent. What about 4? We've got the upper and we have the lower epidermis. And in the lower epidermis we have stomata, and the other 2 cells that control the stoma were guard cells. So they can allow the stoma to open and allow the stoma to close. And then we have our vein, which has xylem and phloem. Xylem is a type of cell that brings water up for photosynthesis to the leaf from the roots, and phloem takes all the sugar produced to all parts of the plant. Okay folks, one last go at this. So you can see the cuticle at the very top, then we have the upper epidermis. And the upper epidermis, these cells are transparent because we need to let light in. Then we've got our palisade mesophyll, full of chloroplasts. Then we have our spongy mesophyll, full of chloroplasts. If we look at the bottom, we've got our lower epidermis, and we can see the 2 guard cells at either side of the stomata. And we can see that they're guarding the stomata. They control the stomata. They can close the pores over, or open them up. Now here we've got the xylem, and that is very important because that allows water to come from the roots to the shoots, and we know we need water in the process of photosynthesis. Now we also need light, and we also need carbon dioxide. So we can see a picture of what might be happening emerging here. Here I can see my light coming in. Remember, the light provides the energy to make your glucose. I can see my water coming in, and I can see my CO2 coming in. And remember the waste product? Oxygen. And the stomata allow gases to come in to the leaf and gases to leave the leaf. Now we're going to look at the chemistry of photosynthesis. So photosynthesis is a series of enzyme-controlled reactions, and it occurs in two stages. Photolysis, which is a light-dependent stage. This does not require enzymes. And carbon fixation, which is a temperature-dependent stage. So we have a stage that's purely about the light and the use of that light energy, and we've got a stage that is using enzymes. Now, the word photolysis, photolysis, photo means light, and to lye something means to split. Now, in this process, light energy is trapped by the chlorophyll, and it's then changed to chemical energy. So light energy is used to split water molecules into hydrogen and oxygen. Now, we've been talking about counting the oxygen bubbles. That's where they come from. That's a waste product. So the hydrogen joins with a hydrogen acceptor. And what we say is reduction means you gain a hydrogen. So this acceptor has gained a hydrogen, so it's been reduced, and that's going to carry the hydrogen to the next stage. Now, you'll be familiar with ATP. That's a high energy molecule that can do work in your cells, and we use the light energy and we change that light energy to chemical energy in the form of ATP, because that's going to help us create the glucose. That's going to help us provide the energy to create the glucose. So, photolysis, what's actually happening? So, light energy splits a molecule of water, and we know that water is a requirement for photosynthesis. Photolysis, I'm using light to split water. And what I get is oxygen as a waste product, and that leaves through the stomata, and we saw that happening. And if you imagine the Elodea, that oxygen will be leaving through the stomata and being a wee bubble that's bubbling up through the water. And we've got the hydrogen. Now we hold on to the hydrogen. An acceptor holds on to that hydrogen for us because we need that for the next stage. Now remember that in this stage I'm also going to require to make some ATP. So I use light energy to join a phosphate onto ADP to make me some ATP, because that's my high energy molecule, chemical energy that can do work, that can do jobs. So from my light dependent stage, which is photolysis, I have got two things. I've got hydrogen, I've got one of my ingredients, if you like, to make glucose, and I have the energy to make glucose, the chemical energy required to make glucose. So let's go through that again. Photolysis, using light, photo, to split. So light energy hits the leaf. Now we know that the upper epidermis are a layer of transparent cells. They allow the light to come in. And we have water in the leaf cells. So the light splits the water into oxygen. H2O is split right down the middle, oxygen is a waste product, and we hold on to that hydrogen. A hydrogen acceptor holds on to that. We need that. That's one of our ingredients to make glucose. But I can't make glucose without energy. So I take some of that light energy and I use that to add a phosphate onto ADP. Remember, ADP is A2Ps. I want to use the light energy as another phosphate on to make ATP. And that's the high energy molecule that I can use to do jobs. For example, make glucose sugar. So let's have a look at the next stage. This is called carbon fixation, and this is enzyme dependent. So it's a series of enzyme-controlled reactions using the hydrogen from the light-dependent stage and the ATP and carbon dioxide from the air. So carbon dioxide plays its role in this stage, and actually the whole stage is named after carbon dioxide, carbon fixation. The carbon dioxide and the hydrogen join together to produce glucose. So CO2 plus hydrogen combined together with help from energy from ATP allows you to make glucose. So carbon fixation. So basically what's happening here is carbon dioxide is going to combine with the hydrogen from photolysis to form glucose sugar. So here we've got carbon dioxide from the air, and that's coming in through the stomata. And we've got hydrogen from photolysis. Remember, we split the molecule of water, H2O. We've held on to that hydrogen. When you combine hydrogen and carbon dioxide together with the help of energy from ATP, because you can't combine two molecules together without energy to do it. Think about making a cake. You might have all the ingredients, but you need to actually physically mix them together, and that takes energy. But when you can join carbon dioxide and hydrogen together, you make carbohydrate, which is glucose. And remember, carbon fixation is enzyme dependent. So that means temperature is important, because we know temperature, obviously enzymes are affected by temperature. We know that low temperatures, enzymes are not working very well. They can be inactive. The optimum is the most active an enzyme is, and at very high temperatures they can be denatured. So just a wee summary here. Photolysis, we're using light energy to split a molecule of water into hydrogen and oxygen. Oxygen is a waste product, and that leaves through the stomata, but we hold on to the hydrogen. We're going to need that. That's one of our key ingredients in making the glucose sugar. We also use light energy to generate ATP. All I'm doing here is changing light energy into chemical energy, because I've got a job to do. The second stage is carbon fixation. They use the word carbon to think, well, carbon dioxide's involved here. And it's enzyme controlled. So carbon dioxide joins with a hydrogen with the help of energy from ATP to form glucose sugar. Now, quite often the SQ might ask you, what is the role of carbon dioxide? What is the role of hydrogen? Well, what the role of something is, what does it join with, what does it form? So what is the role of carbon dioxide would be, well, its job is to join with hydrogen to form glucose. What is the role of hydrogen? Well, its role is to join with carbon dioxide to form glucose. They might ask, what's the role of ATP? And that's always to provide the energy, because that's what ATP does. It's the high energy molecule that does work. What work's it doing? It's providing the energy to join carbon dioxide and hydrogen together to form glucose sugar. Now, what happens to the glucose sugar in plants? Well, a number of things can happen to the glucose sugar. The plant might want to use it up in respiration. So it might be broken down by glycolysis, etc., in aerobic respiration. Or the plant might want to store it it might not need the energy from respiration, so it might convert it to the storage carbohydrate starch. Or it might need to repair damage to a cell wall, and it might change some of the glucose into the carbohydrate cellulose, which we learned about again in Unit 1. Plant cells have cell walls made of cellulose, and that's made of glucose molecules all joined together. So let's remind ourselves of the structure of the leaf again. So we've got the cuticle at the top. What was the first layer called? The upper epidermis. Now, what was special about this layer of cells? They are, remember, the light has to get through, so they are transparent. Okay, the next layer of cells, too. What does a princess drink in her palace? Palisade mesophyll. And then what you might take into the shower with you? Spongy mesophyll. And the palisade and spongy mesophyll are full of chloroplasts, so they're where photosynthesis will be happening. And then we have the lower epidermis, and what's special about the lower epidermis is they have the guard cells and the stomata. And remember, we talked about the vein and having two different types of cells. For example, the xylem cells, which bring up the water for photosynthesis, and the phloem, which transports the glucose that we've made to all parts of the plant. And again, we'll learn about that in the next unit. Now, let's have a look at what happens here. So, we're in the palisade mesophyll here, and we've got water. Okay, now remember, the only cells that can carry out photosynthesis are palisade mesophyll, spongy mesophyll, and the guard cells. So, here we've got light energy. Obviously, it's a sunny day, light's passing through the transparent upper epidermis into the palisade mesophyll. Now, we're going to carry out photolysis. We're going to use light to split H2O, and what we have is hydrogen and oxygen. Now, oxygen is a byproduct. Okay, and that will diffuse out of the plant through that big air space that we can see, because remember, the spongy mesophyll has got lots of air spaces, and the oxygen will leave through the stomata. Now, you'll notice that ATP has been produced in the light-dependent stage during photolysis. We change some of that light energy into chemical energy, which is ATP. If we didn't do that, we might have hydrogen and carbon dioxide, but we couldn't combine them together because we wouldn't have the chemical energy to do that. Now, here we have the carbon fixation stage. Now, carbon fixation, carbon dioxide involved, and the carbon dioxide comes through the stomata. What's the role of carbon dioxide? Well, it's looking for the hydrogen. It's going to join with the hydrogen to form glucose with the help from energy from ATP. Now, what's going to happen to that glucose? It might be carried around the plant by the phloem. So remember, the xylem brings water up to the palisade mesophyll, the spongy mesophyll, the guard cells, but the phloem transports that sugar around the plant. Now we have oxygen leaving. And when we reflect back in the Elodea, the underwater plants, the plants that live in water, if you like, that's where the oxygen bubbles were coming from. So the glucose made in photosynthesis can be used by the plant in a number of ways. It can be used by the plant as an immediate energy source. So that's respiration. It can be broken down. You make it, then you break it down. We take it into our bodies and then we break it down. The plants make it first and then break it down. It can be stored as a complex carbohydrate, starch for example, or it can be used as a structural carbohydrate, for example, cellulose. And we say structural because cellulose is part of the structure of the cell. Now, this is an experiment set up to test leaves for starch. Now, what I'm saying to you is green leaves can carry out photosynthesis. So the hydrogen and the carbon dioxide can combine with the help of energy from ATP to make glucose. And plants will take some of that glucose and change it into starch. Now we've got a test for starch. Can you remember the chemical in labs which tests for starch? Iodine. Now iodine's orange in colour, and in the presence of starch, iodine turns from orange to blue-black. Okay, so if I'm going to test this leaf for starch, I'm looking for a colour change, but the leaf is green, and that's going to interfere with my colour change. It's going to make it hard to see a colour change. So the first thing I'm going to do is heat the leaf. I'm going to cut a wee disc. I'm going to cut a leaf disc using a cork borer, put it in water, and I'm going to boil it. Because what the boiling does, it really bursts the cell wall, it damages the cell wall, it gets rid of the cellulose. And that is important at this stage. And then we turn the heat off, because what we're going to do is we're going to treat the leaf with ethanol and alcohol. Now we know that alcohol and flames don't go well together. Now when we heat the leaf disc in alcohol, what that does is it strips the leaf disc of the chlorophyll colour. So it means you've got a very pale leaf, like a transparent leaf disc, that now we can test for starch. Now, do you remember we get rid of the cell wall by boiling? That allows the iodine to get into the plant cells. So you leave until the green colour is removed completely. Remove leaf, rinse in warm water and place on a tile or dimple tray, and you cover the leaf with iodine and look for the dark areas. No starch there, but plenty of starch there. Now, just go through the procedures. The reason we boiled the leaf was to get rid of the cellulose cell wall, because that allows the iodine to get in. And then remember what we did, we turned off the Bunsen burner because we can't have flames and ethanol, because what we're doing is using ethanol alcohol to get rid of the green colour, so we can clearly see a colour change. That's the only reason we did that, so we can, if we're trying to show a colour change, it's far easier to see when you strip the leaf of the iodine. So factors affecting photosynthesis, there are a number of factors which can affect the rate of photosynthesis, and they're called limiting factors. Now we know this from previously: light intensity, temperature, and carbon dioxide. Now we know a wee bit more about the temperature now, because we know that the second stage of photosynthesis, carbon fixation, is enzyme controlled, and whenever you've got an enzyme working, we know that temperature is going to be implicated. We know that we have to get the temperature to optimum conditions where the enzymes are going to work at their fastest rate. So light intensity, in dim conditions, plants will photosynthesize more slowly than in bright conditions. This is because the more light there is, the more energy is available for photosynthesis to happen. So the more photolysis is going to happen, the more we split the water and the more ATP produced. And we can see this using pondweed called Elodea. So we've talked about this before. We know that if we move the bulb closer to the Elodea, we can see the more oxygen bubbles are released, and we know now that these oxygen bubbles are released through the stomata. Do you remember what the transparent heat shield was for? Remember, the investigation was investigating the effect of light intensity on the rate of photosynthesis. What do bulbs also provide? They provide heat. And if we are looking at changing heat and light intensity, our investigation is no longer fair, our investigation is no longer valid. So the lamp is moved to a certain distance away from the Elodea, and the number of bubbles released per minute is counted. The lamp can then be moved further away and the bubbles counted again. The results would look something like the following. So as we can see, the distance from the plant, as that is decreasing, what's happening to the number of bubbles per minute? It is increasing. Now, What do the number of bubbles per minute represent? They represent the rate of photosynthesis. So I could also say, as the distance from the plant, in terms of the bulb, decreases, i.e. as the bulb gets closer to the plant, the rate of photosynthesis increases. Now we're going to have a wee look at a line graph. So as the distance from the plant decreases, the number of bubbles released increases. So you could also say as the light intensity increases, the rate of photosynthesis increases. So as we can see from the graph, the closer the lamp is to the plant, the higher the number of bubbles. The line levels off, indicating that further increases in light intensity will not increase the rate of photosynthesis. So some other limiting factor must be taking effect at this stage. So remember, if you're on the way up, it's what's on the x-axis. You look down, that's what's affecting the rate of photosynthesis. But when you're on the level, you've got to consider other factors that may affect the rate of photosynthesis. So what would the explanation for that be? When there is a higher light intensity, there is greater photolysis, which is splitting of water, so there's more hydrogen. This means there'll be more carbon fixation, as there'll be more hydrogen for the CO2 to attach to. And also remember, the more light, the more ATP that we produce. So remember, in these investigations, we're only changing one factor, and the factor we were changing was light intensity. So we had to use a heat shield to make sure the only factor we were changing was light intensity, because we know that bulbs give off heat and light. So, carbon dioxide concentration. Now, this investigation also uses the Elodea bubbler apparatus, but this time we're changing carbon dioxide concentration, so every other factor must be kept the same. So we keep the light in the same position, the lamp doesn't change, that stays in the same position. But remember, we used the sodium bicarbonate to give us our carbon dioxide. So what we're going to do is gradually increase the sodium bicarbonate. And what we'll see is the higher the concentration of sodium bicarbonate, the higher the rate of photosynthesis. Because the more carbon dioxide, the more CO2, carbon dioxide, will join with hydrogen to form glucose. Now obviously that will eventually level off because some other factor will become limiting. Temperature. Temperature is also a limiting factor for photosynthesis. Now, light-dependent stage is not affected by temperature, but carbon fixation is. And because enzymes, it's all about the enzymes and carbon fixation, the graph is not going to be the same. We are looking back at our enzyme temperature graph. Now we know that enzymes are affected by temperature. At low temperatures, enzymes are inactive. At optimum temperatures, they are their most active, they peak, but at high temperatures, they are denatured. So we're not going to get that same graph. We need to keep the temperature at optimum. And this is the graph that you would see. So as the temperature increases, the number of bubbles would increase and then would decrease. Because at low temperatures, the enzymes would be inactive. At optimum temperatures, they'd be most active, and at high temperatures the enzymes would be becoming denatured. We can see from the graph that as the temperature increases, so does the rate of photosynthesis. This happens until it reaches its optimum temperature. After this, the rate of photosynthesis slows down as the enzymes which control it are denatured. Okay folks, so lettuces may be grown in tunnels covered in clear polythene as shown in the diagram below. We've seen this before. Now, basically this polythene increases the temperature for photosynthesis. Now we know that the second stage, the carbon fixation stage, requires enzymes. So if we're making sure the enzymes are working at their optimum temperature, we're going to get a faster rate of photosynthesis and larger lettuce plants. Now, think, pair, share. Try to work out what this pupil is trying to find out. What do you think? What are they trying to prove? What are they trying to investigate? So if you look at leaf A, we've got leaf A in an airtight glass tube, and it's including a substance which absorbs carbon dioxide. So if you're absorbing carbon dioxide, you're removing it. So leaf A will not be receiving any carbon dioxide. So what is this pupil trying to find out? Is carbon dioxide required for photosynthesis? Now leaf B would be a control. It'd act as a comparison. Leaf B would be able to carry out photosynthesis, but leaf A would not, proving that carbon dioxide is necessary. Now using this information, try to work out how you would redesign this experiment to find out if light was required. What might you do to one of the leaves? What you could do is cover one of the leaves with black card and have B as a comparison to prove that light is required. Now we're going to look at respiration versus photosynthesis. Now, we know that green plants carry out photosynthesis and they carry out respiration. We know that animals carry out respiration. We know that respiration and photosynthesis are controlled by enzymes, and we know that enzymes are most active at optimum temperatures. So, we're going to work out which processes are happening in each of these test tubes. So we've got tube 1, 2, 3 and 4 and a green plant in each one. And we've got to decide whether respiration, photosynthesis, both or neither are happening. Now, variegated means you've got green, you've got chlorophyll and a bit of white. Okay, so remember that green plants carry out photosynthesis and they carry out respiration as well. And remember that both processes are enzyme controlled. All of respiration is enzyme controlled and carbon fixation is enzyme controlled. So temperature is important. I'm going to give you a couple of minutes to think about it. We'll regroup and we'll go through each one. Now, I should say that every single tube contains water. So tube 1, we've got a black cover. We've got the test tube and a black cover. What processes do you think will be happening? So let's think about photosynthesis. Can photosynthesis happen in the dark? What do we need for photosynthesis? Well, you should know by now that we need light, so photosynthesis will not be happening. Let's think respiration. Do we need respiration? Do we need light for respiration? Human beings respire. When you go to sleep at night, do you stop respiring? No, so it doesn't matter. Respiration doesn't matter whether it's light or dark. Respiration will still be happening, but photosynthesis will not. Let's look at tube 2. We've got a green leaf and water. Well, can photosynthesis happen? Is there anything stopping it from happening? Well, light can get at the leaf. We've got water. So photosynthesis will be happening. What about respiration? Is there anything stopping respiration from happening? No. So both will be happening in tube 2. Let's look at tube 3. We've got a boiled green leaf. Boiled. What does boiling mean? What have we done to the leaf? If you boil a kettle, what have you done to the water in it? You've heated it to very high temperatures. What do we know about photosynthesis and respiration? What are they controlled by? Enzymes. What do we know about enzymes? Do they like to be boiled? So what process will be happening in tube 3? Neither. Now tube 4, will photosynthesis be happening? Remember, variegated means we do have chlorophyll, but maybe a bit less. So there's nothing to stop photosynthesis from happening, but maybe not as much as tube 2, for example, which is not variegated, which is just green. What about respiration? Is there anything stopping respiration from happening? No. So, both. Okay, so again, on the comparing respiration and photosynthesis route, here we've got a list of carbon dioxide, cellulose, glucose and starch. We have to complete the following table with the correct carbon compound for each of the functions. So, what would our raw material for photosynthesis be? What is something that we know we need to carry out photosynthesis? So we should know that in carbon fixation we need carbon dioxide. What about a respiratory substrate? Now, what does substrate mean? Substrate is the molecule you start with. So if I'm asking you, what do we start with? If I was to ask you, right, let's do an essay on respiration, what's the first word you write down when you're going to do, for example, glycolysis? What are you breaking down into pyruvate? You're breaking down a molecule of glucose. So glucose is your respiratory substrate. That's what you start with when you're carrying out respiration. And what would a storage carbohydrate be? Now, remember when we did photosynthesis, the plants obviously combine hydrogen and CO2, carbon dioxide, to form glucose. But they don't use all that glucose up in respiration straight away. Sometimes they might store it as starch. Now we're going to have a wee look at a photosynthesis essay. So we're going to have to write notes on the stages of photosynthesis. Now I'm going to give you a couple of minutes to have a think about what you might write down. Again, don't worry if you get this wrong. This is just grab a bit of paper, grab a pen and write down what you've remembered so far. So what you might have is that light energy from the sun is trapped by chlorophyll and converted to ATP. You might have said that water is split by light energy to produce hydrogen and oxygen oxygen diffuses from the cell. You might have said that carbon fixation is when carbon dioxide combines with a hydrogen using energy from ATP to form glucose. And you might have said that carbon fixation is controlled by enzymes. Okay folks, we're going to do some exam style practice now. So the light energy for photosynthesis is captured by chlorophyll contained in chloroplasts. Okay folks, what is the name of the first stage of photosynthesis? So we should have photolysis, using light to split a molecule of water into hydrogen and oxygen. And remember, we also use the light energy to make ATP. What diffuses out of the leaf? Oxygen, and that's the waste product. And what's two products used in the second stage? Two products produced from the light-dependent stage, from photolysis, used in the second stage? Hydrogen and ATP. Okay folks, so the graph below shows how light intensity affects the rate of photosynthesis under different conditions. So remember, when you're on the way up, you look down, and if you're on the level, you look up and spot the difference. And whichever is different and higher is what's affecting photosynthesis. So let's look at point A. What's affecting photosynthesis at point A? So obviously we're going on the way up, so we look down, so that will be light intensity, B. So we're on the level, so you look up and spot the difference. So what's higher? CO2. So what's holding it back, what's limiting photosynthesis at B is CO2 concentration. Let's look at C. Look at the level above and spot the difference, and it is temperature. There's a higher temperature above. Okay, the graph below shows the effect of temperature on the rate of photosynthesis in a green plant. A correct conclusion would be that so what's happening as the temperature increases? The rate of photosynthesis is increasing. An experiment was set up to measure the effect of light intensity on the rate of photosynthesis in the water plant Elodea. The light intensity was varied using a dimmer switch on the bulb. The rate of photosynthesis was measured by counting the number of bubbles released per minute. What would the gas collected be? So what gas leaves through the stomata? Oxygen. And remember, oxygen is formed by water being split into hydrogen and oxygen. So, an experiment was set up to measure the effect of light intensity on the rate of photosynthesis in the water plant Elodea. So we've got the experiment set up here. Suggest how the investigation could be improved to give more accurate results. So we're looking at our measurements. How could we measure the oxygen more accurately? We could use a measuring cylinder to collect the gas to actually get a measurement of the volume. Rather than just counting bubbles, we'd use the volume collected over time. There are two reactions in photosynthesis. The first reaction is photolysis. Name the two substances produced by photolysis that are required for the second reaction. So what is produced when you use light to split water that's required for the next stage, and what else do we use the light for? So we should have ATP and hydrogen. Name the second reaction. What's the second stage called? Remember, carbon dioxide is involved, so it is carbon fixation. Plant cells convert glucose into other carbohydrates. Complete the table below by naming two of these carbohydrates. So which one is a storage molecule? So the glucose produced won't all be used in respiration, but some will be stored as starch. And again, not all of the glucose used will be used in respiration. Some will be to repair cell walls, which are made of cells. So the diagram below shows an investigation into photosynthesis. So which of the following statements is correct? So as you can see from the diagram, we've got P, R and Q. These leaves have been treated differently. So Q is covered in black paper. P has got a carbon dioxide absorbing powder. Now if you absorb carbon dioxide, that means you remove it. Okay, and R has not been treated. So which of the following statements is correct? Which ones are you going to make food? Which ones are you going to be able to carry out photosynthesis? Remember what you need for photosynthesis, and if you take that away, it won't happen and you won't make food. So you should have only R, because P you've taken away the CO2, and Q you've taken away the light, both required for photosynthesis to make food. The role of chlorophyll in photosynthesis is to trap. So we should have light energy to make ATP. The raw materials for photosynthesis are, what two things do you need? We need carbon dioxide and water. Most plant cells have a cell wall. Name the structural carbohydrate in the cell wall that the glucose molecules can join together to make. Cellulose. Photosynthesis is a process by which green plants make glucose using energy from the sun. Name the byproduct A released during photosynthesis. Oxygen gas, and remember it leaves through the stomata. Hydrogen and a high energy molecule are produced during photolysis. Name this high energy molecule that's produced from the light energy. ATP. Describe the use of hydrogen in carbon fixation. So that is what's the role of hydrogen. And if you remember what I said is, the role of something, its use is what does it join with and what does it form? So hydrogen joins with carbon dioxide to form glucose. Now you'll notice that to form glucose on its own it's wrong. However, if you say what it joins with and what it forms, that would be correct. Explain why an increase in temperature can lead to an increase in the rate of photosynthesis. What do we know about the second stage of photosynthesis? Well, we should know by now that it is controlled by enzymes. Now, we've done the enzyme topic and we know that enzymes are affected by temperature. So, temperature affects the activity of enzymes and enzymes affect the rate of photosynthesis. The graph below shows the effect of temperature on the rate of photosynthesis in a green plant. What would a correct conclusion be? What's happening to the rate of photosynthesis as the temperature increases? Well done. As the temperature increases, the rate of photosynthesis also increases. And obviously we're looking at between 10 and 15 degrees. We're obviously looking for the plant to reach optimum temperature for optimum photosynthesis. So other than temperature, state two limiting factors of photosynthesis. So what two things do we need for photosynthesis to happen? And if they're not there, it won't happen as well. So we should have light intensity and carbon dioxide concentration. So the graph shows the effect of varying the light intensity, temperature and carbon dioxide concentration on the rate of photosynthesis. So remember our rule, if you're on the way up you look down. So what's limiting photosynthesis at S? So we should have light intensity, so that rules out A and C. So let's look at T. We can see that we're on the level, so we look up and spot the difference. So B. Which of the following conditions in a greenhouse would produce earlier crops? So again, you're looking for something that's going to increase the rate of photosynthesis. What do you need for photosynthesis? Carbon dioxide. The graph below shows the effect of two different environmental factors on the rate of photosynthesis. What is limiting the rate at X? Remember our rule? You're on the way up, you look down. What's on the x-axis? Light intensity. At Y, when you're on the level, you look up and spot the difference. Now look at the key to the right. What has increased at the level above that's affected the rate? Carbon dioxide concentration. State one way that the rate of photosynthesis can be measured. Remember the Elodea. How do we measure the rate? Number of bubbles, or even better, using a measuring cylinder to measure the volume of oxygen over time. Now we can't say just a number of bubbles or volume of oxygen because it's rate, and rate is over time. During the first stage of photosynthesis, light energy is used. Where is light energy trapped in the cell? What is it plants have that allows them to trap light energy? They have chlorophyll or chloroplasts. State one use of that light energy. Think about the light dependent stage. There's two options. So it can be used to split water or used to form ATP. Name the second stage of photosynthesis. Remember, carbon dioxide is involved here, so we call it carbon fixation. Name the carbohydrate produced during the second stage of photosynthesis. When we join carbon dioxide and hydrogen together, what do we get? Glucose. The sentence below gives some information about photosynthesis. You've got to work out what words would make that sentence correct. So, photosynthesis uses what's required? Carbon dioxide. Now we know that. We know that a whole stage in photosynthesis is named after carbon dioxide, the carbon fixation. So photosynthesis uses carbon dioxide to allow green plants to make their own food, and some of this food is converted to cellulose for making cell walls. Now some of the food is converted to starch, but not for making cell walls. Remember, starch is a storage carbohydrate. So the first reaction in photosynthesis is called carbon fixation. Is that true or false? So we should have it's false. What would the correction be? What's the first stage called? Photolysis. Hydrogen is transferred from the first reaction to the second reaction. Do we hold on to the hydrogen? We do, so that has to be true. And ADP is used as the energy source for the second reaction. And we should know that that is false. It is ATP. So again, another limiting factor graph. What would be limiting photosynthesis at P, Q and R? Remember, when you're on the way up, if you're going up, you look down. If you're on the level, you look up and spot the difference. So P, that has to be temperature because that is on the level. It's not what's on the x-axis, because as I increase or decrease carbon dioxide concentration, the rate stays the same. So we look at the level above and spot what's different, and what's different is the temperature. So temperature is holding the rate back. Then we're looking at Q. Now Q's going up. We're on the way up, so we simply look down, and it's whatever is on the x-axis, which is carbon dioxide concentration. Now we look at R. Now remember, we talked about R. If you don't have a level above, you've just got to think, well, what else can limit photosynthesis? So it might be light intensity. Or you could say we could go up a wee bit higher in terms of the temperature. But it can't be what's on the x-axis. So if we're level, it can never be what's on the x-axis. So what is stage 1 called? So stage 1 is called photolysis, and stage 2 is carbon fixation. Okay folks, here's a wee mini essay question. Describe the transfer of energy from light arriving at the leaf to the formation of sugar. So it's asking about photosynthesis. So there's a lot of things we can say. I want you to think about it for a few minutes and we'll have a wee chat about it. So what you might say is, light energy is trapped by chlorophyll. Light energy is used to split water into hydrogen and oxygen, and you might want to add this is called photolysis. You could also say that oxygen is released and hydrogen is used in the second stage. Now we also want to see that light energy is changed into ATP in the light reactions as well. And ATP is transferred to the second stage. And in the second stage, which is called carbon fixation, hydrogen and CO2 combine to form glucose with the energy supplied from ATP. And you could also say this is called carbon fixation. The rate of photosynthesis can be affected by several factors. So again, what you have to do is select the word that makes that sentence correct. The limiting factor is the factor which is preventing the rate of photosynthesis from, unless the supply of that factor. So again, we've got the same kind of question. When we're on the way up, we look down, and if we're on the level, we look up and spot the difference. So what is affecting or limiting the rate of photosynthesis at X and Y? So we should be quite good at this by now. So for X, we should have carbon dioxide because we're on the way up, so we look down, and with Y, we look up and spot the difference. Okay folks, you have to select from the words in the bank and bring them down in terms of the carbon compound that matches the function. So what's a raw material for photosynthesis? What do we need for photosynthesis? So that would be carbon dioxide, and a respiratory substrate would be glucose. We talked about this. What is used, what's broken down in respiration, and cellulose is a storage carbohydrate. Okay, so we're quite familiar with this apparatus. The apparatus shown below is used to measure the rate of photosynthesis at different light intensities by counting the bubbles of oxygen released. Now we know why the oxygen is produced, because light energy splits water into hydrogen and oxygen, and obviously the oxygen is released. So if we're investigating the effect of light on photosynthesis, how could the light intensity be varied to allow us to do that? Well, we'd move the lamp closer or further away. Now, why would the level of dissolved oxygen in the water fall after the lamp was turned off? Now, this is quite a tough question, because we might expect, if we're just talking about photosynthesis, if we switch the lamp off, we'd no longer produce oxygen because we need light for photolysis to give us oxygen as a byproduct, but I wouldn't expect it to fall. If it was falling, it means it's decreasing. So why might oxygen be used up? What other process do plants carry out that might mean that oxygen actually starts to fall? Respiration. Name the substance found in chloroplasts which captures the light energy required for photosynthesis. Chlorophyll. Name the substance formed by plants to store glucose produced during photosynthesis. Starch. It's a storage carbohydrate. Quite an easy one. What's limiting photosynthesis at X? Remember, if you're on the way up, you look down, and what's down is light intensity. Okay, which of the following statements is true? A, B, C, D. Now we've done this one before, so it should be quite easy. Which 2 out of the 3 are not going to be able to carry out photosynthesis and produce sugar? So what's the only one that is? D. Okay, then the pigment and chloroplasts which trap the light energy produced. Chloroplasts contain chlorophyll. In the light-dependent stage of photosynthesis, light energy is converted to chemical energy. Identify substance A in which the chemical energy is stored. ATP. Identify substance B, which is produced in stage 1 and required for stage 2. So look, we've got water being split, we've got a byproduct. What would substance B be? Hydrogen. State 1 substance into which the sugar produced in stage 2 can be converted. So we talked about glucose can be formed as sugar, but we don't just use all the sugar in respiration, it can be changed to other things. Starch, which is storage, carbohydrate, or cellulose, which is structural, found in cell walls. Explain why temperature can limit the rate of photosynthesis. What do we know about the second stage of photosynthesis? Well, we know it's controlled by enzymes. Right, so if it's controlled by enzymes, we know that enzymes are affected by temperature, and the lower the temperature, the lower the activity of the enzymes. Okay, so photosynthesis is a two-stage process. We have light reactions and carbon fixation. The table below shows some statements about photosynthesis. So you've got to tick which ones are true. So for stage 1, the first couple are given. Is water required for stage 1? Photolysis, that would be correct. Is it required for stage 2? No. What about sugar produced? What stage is sugar produced? Glucose produced? Stage 2. ATP and hydrogen required, not made, required. So they are made in stage 1, but they're needed in stage 2. And in what stage is oxygen produced as a byproduct? Stage 1, because oxygen is produced from light energy splitting water. Explain why high temperatures above 50 degrees Celsius would prevent the photosynthesis reactions from taking place. And again, what do we know about photosynthesis, particularly carbon fixation? Photosynthesis is controlled by enzymes for carbon fixation, and at high temperatures we know that the enzymes are denatured. State 2 environmental factors which could limit the rate of photosynthesis between point P and Q. So again, when you're on the way up, you look down, but here between P and Q we're on the level. So it's not what's on the x-axis. So it's some other factor. So it can't be carbon dioxide. What else could be holding back the rate of photosynthesis? What other factors affect photosynthesis? Light intensity or temperature. Remember, when you're on the level, that is not what's on the x-axis. When you're on the way up, it is. On the level, it's not.